Hi y'all, it's Miss Barker here. I want to spend some time today talking to you about the scientific method. This is going to be a super brief overview because you should have talked about this in numerous science classes before. I just want to do a quick refresher. First, what is the scientific method? So we're going to use it as a series of steps to investigate a problem. And we're going to talk about each of these steps individually, but I want you to make note that this is not a linear prescribed method. When I say steps, they don't necessarily go in order the same way every single time. Instead, it's more of a process, and we can adapt that process to the needs of our specific experiment. So here's what our general process is going to look like. We're going to identify a problem, collect information about that problem, form a hypothesis, develop and test an experiment, accept or reject our original hypothesis, and report our results. Two extra pieces that really need to be mentioned here. If we accept our hypothesis, we're going to go back and retest it again, probably multiple times if we're, following best practices. And that's because we want to confirm our results. We want our results to be as strong as possible. We do that by confirming them multiple times. If we decide to reject our hypothesis, then we need to take a look at that hypothesis and kind of tweak it, see what we can change, and test it again. Okay. First step, identifying a problem. So it's important when we are identifying a problem that it is very, very specific. We want our problem to be well defined, and we want it to look at one particular variable that we can change. If our problem is too broad, then we can't test it, and that makes it kind of useless for us. So if we do have a very broad problem we want to work towards, we can break that down into several smaller individual pieces that we can build on each other. Next, we are going to collect some information about our problem. We're going to do some research. The important thing here is that we are gathering our research from reliable resources. If your original information is not coming from a reliable source, it's not a great thing. It's not going to be super helpful when you're designing your experiment. So how do we know when a source is reliable? Best way, best practice, is to use peer-reviewed articles. Part of the problem with this is that sometimes they are behind a paywall, meaning that you have to pay to access the article. That's kind of frustrating. I get it. Researchers have to eat too. But it can be an annoyance for us. Additionally, a lot of these papers are super, super technical. They are written at a really high level with really specific language to whatever field they're written in. That makes it kind of inaccessible to anybody who's not actively working in that field. So to sum that up, peer-reviewed articles are the best. They are the gold standard when we are looking for research. However, it's okay that we might have to sometimes use additional sources to help us fill in the gaps if we are not researchers in that field. A good way to find peer-reviewed articles is to use Google Scholar. So Google Scholar does a really great job of compiling peer-reviewed articles from lots of different publications in one place. That's a great place to start. So if we can't find a peer-reviewed article or it's inaccessible to us, we may have to use a web page. That's fine. We just need to make sure that the web page is reliable. So first we need to ask ourselves, who's in control of this information? .gov sites are usually pretty good and reliable. They're a good place to start. One of the positives about using .gov sites is that they cite their research, so you can often go back and find peer-reviewed articles from there. If a corporation is running the website, it's important to know what their interest in the topic is. Similarly, was the study that they're referencing paid for by somebody with a vested interest? So how was the research funded is a really important question to ask. Once we have identified our problem and we have done our research, it is time to form a hypothesis. So a hypothesis is a testable, specific solution to our problem, or answer to our question. So if I want to look at how sunlight affects flower growth, then my hypothesis may be something like increased exposure to sunlight will cause flowers to grow taller. That's a hypothesis. It's specific, it's testable, it addresses my question. Now we can design an experiment to test our hypothesis. If you get to this step and realize that you have no idea how to do that, go back and double check your hypothesis. Make sure it's actually testable. Make sure it's something that you have a way to design an experiment around. In order to do that, we need to look at some variables. So variables are just any factor in an experiment that can change. In my example, I have lots of variables. I have the amount of sunlight. I have the height of the flower. I have what type of food I'm feeding the plant, how much food I'm feeding the plant, what type of soil it's growing in, what type of climate it's in, the humidity and temperature, the time of day that I'm taking the measurements, all kinds of variables. So ideally, we want to have one independent variable and one dependent variable. So the independent variable is going to be the one that we are purposely changing. It's the one that we think is going to cause the effect we want to see. So in my example, this would be the sunlight. The dependent variable is the variable that you expect to change as a result. So you're not actively changing it, it's changing in response to the independent variable. In this case, that would be plant growth. All other variables are considered controlled variables. You're going to keep them the same between all of your testing groups. So what is a control group? A control group is where we have no variables manipulated. It's our baseline. This is important because it allows us to compare the experimental groups to something. We can compare them to what would happen if we had no intervention, no change in our independent variable. This can help eliminate the effects of confounding variables. So those variables that we talked about, the other variables that may affect plant growth, there are sometimes things that you can't necessarily hold constant. You can't hold the weather constant. You can't always account for fluctuations in, like small fluctuations in temperature. So having a control group helps you eliminate the effect of those small changes, because anything that's happening to your experimental groups is also happening to your control group. Once we have done our tests, it is time to accept or reject our hypothesis. So you're going to determine if your results support your original hypothesis. Is there evidence that your experiment shows what you thought it would show? If not, you're going to reject your hypothesis. You're going to say, nope, this wasn't it. That may lead to more research. You may go back to searching for other sources to see if there was something you overlooked, if there was some other influence that may have led to the result you saw. Or you may just go back and amend your hypothesis. You may say, okay, well, we weren't quite right the first time, so we're going to tweak it a little bit to this and retest. Now, it's always important, even if you reject your hypothesis to report your results. Super important to report. And that's because somebody else could potentially see your results, and maybe they were thinking along the same lines, and that gives them somewhere to start from to develop their own hypothesis. So take a few seconds and think about what are the minimum requirements for something to be a scientific process. So either you just watched it or you're about to. We're going to do an activity with a video that is not from a scientific field, but the subject goes through a very scientific process. So think about, what exactly is it that makes something science? Is it the technical vocab? Is it the hypothesis making? Is it the way you design your experiment? What is it that a scientific process needs to have?